Good afternoon to everybody. I am Gianni Capasso, and together with Marion Pepin, my co-chair, we will have the fortune and the honor to chair these pre-Congress meetings of the 59 ERA Congress in Paris. You know, this is a new introduction by the ERA Society, and the importance of the team is very underlined by these selections, we can say. You know, the team, the objective of this symposium is to analyze the importance of electrolytes on brain function. And as you know, in the last couple of years, ERA has paid a large attention on the effect of brain function, of kidney function on the brain function. This is and today we will address an important issue: how electrolytes works on the brain function. And to deal with this aspect, three outstanding speakers have been invited. Carsten Wagner is a physiologist from Zurich. Julia Refardt she's an endocrinologist from Bern, and Pedro Imenez Silva is again a physiologist, but a Brazilian physiologist working in Zurich. And I will have the honor to introduce, first of all, Carsten. Carsten Wagner, as I say, is an outstanding researcher. He's a professor of physiology in the University of Zurich, and is well known worldwide for his studies on several aspects of the kidney function, and in particular on the phosphate metabolism and phosphate handling by the kidney. And I have the honor now to ask Carsten Wagner to give his talk on the phosphate and the brain. That is a new issue in the brain physiology and also in kidney physiology. Carsten, please start with your talk.
Oh, thank you very much for the kind words and invitation. What I would like to do is to talk about a few aspects how phosphate and the brain interact. And I have to make a few disclosures here. And I would like to start here with this old advertisement for a phosphate-containing drink called Horsford's acid phosphate. As far as I know, it's not available anymore, so I can make an advertisement for this. And the old advertisement stated that this drink would be particularly good if you had mental exertions and exhaustion. It would increase your intellectual power, and only 5 drops in a half glass of water would give you excitement and would increase your intellectual fitness. I think this is very promising. Now there is a more modern product that I found that apparently enhances memory function, and it contains various forms of phosphate. So these are very promising claims here, and I would like to see with you whether any of these claims are really true. Now, we all know that phosphate is critical for life. It is part of DNA. It forms membranes. It's important for signaling events. It's part of ATP, our energy source. And of course, it gives stability to bone. Now, being an essential mineral, we take it up from the diet. It redistributes in the body. And we all know as nephrologists or kidney physiologists that the kidney is, of course, the gatekeeper of phosphate balance, as the kidney has to match the daily uptake with an equal amount of excretion of phosphate into urine. And we also know that in patients with kidney disease, this mechanism fails and leading eventually to phosphate overload. Now, in a healthy individual, of course, we have an intricate system that helps to maintain phosphate balance. And we particularly have two hormones, a parathyroid hormone and a fibroblast growth factor 23, that increase in response to phosphate intake and help the kidney to excrete phosphate, while in states of low phosphate availability, the kidney produces calcitriol, and that helps the intestine to absorb more phosphate and again restoring phosphate balance. And these three hormones actually are really linked to each other in an intricate network where they control each other's levels through positive and negative feedback loops, making the whole system very balanced, but also very complex. Now, how does phosphate act on the brain, and how are some of these hormones involved in shaping brain function and actually also brain morphology? Now, this is very recent data that demonstrate that phosphate acts on the autonomous nervous system. And these are data here from rats that indicate that an increase in phosphate intake increases sympathetic nerve outflow. And we all know, of course, that sympathetic nerve outflow regulates cardiovascular functions, but it also regulates the ability of the kidney to maintain salt homeostasis, water homeostasis, and eventually to regulate blood pressure. And so it's maybe not surprising that Reto Grapf and colleagues showed in young healthy volunteers that a sustained increase in phosphate intake leads to an increase in systolic blood pressure. This is a small increase of only 4 millimeters mercury. But if we consider that this is about the same, we can lower blood pressure by giving thiazides. So I think this is a substantial increase here in blood pressure. And this was associated with an increased activity of the sympathetic nerve system, as indicated here by higher levels of metanephrine and normetanephrine in urine. Now, are there pathologic impacts of phosphate on the brain? Now, sometimes rare disease can help to elucidate underlying function, underlying effects. And there is particularly one disease that is associated with deposits of calcium phosphate in the brain, which is called basal ganglia calcification. Now, this is actually a relatively frequent incidental finding in CTs and can be found up to 1% of cases. And it is not really pathologic unless it is found in patients below 40 years. Then it's worth to be further diagnosed. Now, often it's actually asymptomatic. Then again, there's no need to do anything. But if symptomatic, it can have variable symptoms that include motor deficits and also psychiatric effects. Causes for this can be after various types of infections, intoxications, and then there are rare cases like in inborn forms of hypo and hyperparathyroidism, and also in a disease called FAST disease. Now, FAST disease can be caused by various genes, and two of them are directly related to phosphate homeostasis. SLC20A2, which is a phosphate transporter, which is expressed in the choroid plexus in the brain, and another protein, XPR1, that is linked somehow to phosphate metabolism, but we do not really know what its exact function is. Now, in knockout animals for this phosphate transporter, we can observe that the phosphate content in the cerebrospinal fluid is elevated, and this probably triggers then these calcium phosphate deposits. Now, let's look first at states of hypophosphatemia and then later at states of hyperphosphatemia, how they act on the brain. Now, hypophosphatemia has a variable incidence depending on various groups of patients. And you can see it ranges from a large general hospital patient collective with about 2% up to 80% in patients with specific diseases, such after a liver surgery and so on. So it is there is some prevalence, and it's certainly worth to be looked at. But we should also remember that often hypophosphatemia remains asymptomatic unless plasma phosphate levels fall really very severely below 0.3 millimoles per liter. And then multiple organ systems can be affected, and probably a common denominator of all these symptoms is that hypophosphatemia, such severe hypophosphatemia, leads to intracellular ATP depletion, so there is energy depletion, and it affects also the capacity of red blood cells to transport and release oxygen. So in states of low phosphate availability, hemoglobin has a higher affinity to oxygen, and the release of oxygen into tissue is reduced. Now, I mentioned there are several organ systems that are affected, and I'd like to focus here only on the neurological effects. Again, we find neuropsychiatric symptoms like exhaustion, depression, and in very, very severe cases, this can lead to tremor, seizures, and even coma in these patients. Now, there have been a number of studies looking into phosphate metabolism and energy metabolism in the brain in states of hypophosphatemia, but also in states of primary brain disorders. And this is one of these studies using NMR technology, and they compared 15 control patients, control subjects with 15 subjects that had mild cognitive impairment and 11 patients with incipient Alzheimer's disease. And they all had already reduced cognitive capacities. And what they found using this technology is that there was a reduction in phosphate-related energy metabolism in the brain, and this was mostly attributed in the discussion to impaired mitochondrial function. Similar effects were seen in another study where patients with Alzheimer's disease were compared to age-matched healthy controls. And unfortunately, we don't have in this study any information on plasma phosphate levels or kidney function. But again, what was reproduced here is that there is evidence for energy depletion in various regions in the brain. And again, some of these studies have been even replicated in additional studies in patients with HIV neuropathy or patients with different forms of schizophrenia. So there is evidence that some disorders that primarily affect the brain are associated with altered energy and phosphate metabolism. But now, what happens if hypophosphatemia is the primary disorder? Is this associated with brain dysfunction? Now, this is again a purely observational study, and here 109 patients with mild cognitive impairment were compared with 73 patients that had a mild form of Alzheimer's disease, and several imaging technologies were used to measure amyloid depositions in the brain, and this was associated with plasma phosphate levels. Unfortunately, we don't have a control group here, but what they found was that the lower the serum phosphate levels, the higher the amyloid depositions in the brain. So this might be a pure and coincidental finding. It's a relatively small cohort, but it's certainly worth to be followed. Now let's look now at the other side of the spectrum in hyperphosphatemic states. And we all know that patients with end-stage kidney disease or advanced stages of chronic kidney disease have higher phosphate levels, and that these higher phosphate levels really associate with survival and morbidity and mortality in these patients. And there are numerous studies that have shown this. Now, this is to some extent also true for the general population that is having normal kidney function. Also there we find an association between serum phosphate levels and the incidence of cardiovascular disease, here shown as incident heart failure, and even overall mortality associates with serum phosphate levels in a general kidney healthy population. So this has really led to the concept of phosphate toxicity as here in this recent review. And you can see that phosphate toxicity affects many different organs, but we're all wondering, what about the brain? The brain is missing here on this scheme. Now, there are a few preclinical studies in mouse models where animals were fed a high phosphate diet early on after weaning. And interestingly, this leads to a reduction in brain weight and various alterations in transcription and translation of key proteins in the brain. Unfortunately, we don't have any information whether this has also any functional relevance in these animals. And of course, even further, would this have relevance in children with end-stage kidney disease where hyperphosphatemia may occur while the brain is still developing, or in mothers with end-stage kidney disease and during pregnancy? Could that impact on the unborn child? I think these are interesting questions and require further investigation. Now, in patients with on hemodialysis, the issue of the impact of hyperphosphatemia on the brain has been further investigated, and it was found that there's indeed an increased hazard risk for developing brain hemorrhages in patients with the highest quartile of phosphate levels. But surprisingly, this did not associate with a higher risk to have brain stroke. On the contrary, the group with the lowest phosphate level had the highest hazard ratio for stroke in this study here with almost 3,500 patients. Now, this relationship between serum phosphate levels and stroke risk has been further analyzed in various studies. And this has led to a very recent meta-analysis that included at the end 5 clinical trials with over 32,000 patients. And looking at the relationship here between phosphate and the relative risk, no association was identified. So it seems that if at all, this is a very weak association between phosphate levels and the risk to develop stroke. However, what has become evident in more recent studies is that the outcome from stroke is modified by phosphate. And this is a U-shaped curve here, as we can see for various parameters here. So the group with low phosphate as well as the group with high phosphate serum levels has a worse outcome after stroke. It remains unclear what mediates these associations here. Now, phosphate, of course, when changing, is not acting alone on the brain. And I've shown you various hormones that are involved in the control of phosphate homeostasis. And I'd just like to summarize this here on this slide. There are now more and more data coming out that these hormones per se might act on the brain. So calcitriol, for instance, impacts on the pre- and postnatal brain development in animal models. Again, in an animal model, the hypervitaminosis D that is present in animals that lack cloto and have very high FGF23 levels, this causes motor disease, and reducing vitamin D levels restores motor function in these animals. In human patients, as well as in animal models, there are various associations of vitamin D and vitamin D receptor variants with Parkinson's disease and its severity. Cloto, this cofactor for FGF23 signaling in the brain, is expressed in the brain. It seems to have neuroprotective functions there. It seems to protect from cognitive decline and aging-associated changes. And in animal models, it modulates resident immune cells. In animal models, as well as in patients, high levels of parathyroid hormone in primary and secondary forms of hyperparathyroidism affect mood, as well as cognitive functions in patients. And very importantly, this is to a good extent reversible when PTH levels can be normalized. And it seems to be independent from calcium and phosphate levels that are, of course, modified by PTH. So to summarize, we have certainly some evidence for interactions between phosphate and phosphatemic hormones and brain functions, but there are a lot of open questions. It seems that there is an association of low plasma phosphate levels with higher amyloid deposits in brain, and how this is linked, it remains unclear. There is clear evidence for altered phosphate and energy metabolism in primary disorders affecting the brain, such as Alzheimer or mild cognitive impairment. In patients with chronic kidney disease and high plasma phosphate levels, this associates with a higher risk to develop brain hemorrhages, but it's not associated with a higher risk for stroke, despite the well-known association of high phosphate with cardiovascular morbidity and mortality. Both low and high phosphate associate with worse outcomes after stroke, and maybe this is just a surrogate marker for otherwise a poor condition of the patient. We see that phosphate regulating hormones impact on brain function and in various domains. And it remains unclear whether phosphate itself impacts on brain cognitive functions in the context of chronic kidney disease. And I think it's very clear we have a lot of patchy data, but what we really need is here better and more preclinical and clinical studies to address many of these issues. And with that, I would like to thank to acknowledge also the Connect network, where some of these issues are now being addressed. Thank you very much for your attention.
Thank you very much, Carsten, for this very nice presentation. I understand that the subject of your talk is quite new, quite complex, but I think is a good start in order to, to highlight the importance of phosphate in brain function. This means, in other words, the importance of the kidney on the brain function. I urge all the audience, if they have any question, to use the question and the response mode. And I see there is already one question direct to Carsten. A colleague of us is asking if is low phosphate level reflecting malnutrition and inflammatory status increased risk?
I think this is one of the open questions indeed. I would suspect that indeed hypophosphatemia is an indicator for some other underlying processes. And I've shown this wide association of various clinical conditions with hypophosphatemia. So I would indeed think it is just a surrogate marker for another underlying condition.
Yeah. And if I may put just one question, you know that actually there are very strong data that correlates kidney function, the decline in kidney function with the decline of brain capacity in terms of cognitive capacity. And we always think that PTH and the phosphate, all the other, may be in somehow very well connected. Do you think there is a space for clinical study in addition to preclinical studies to try to clarify these evidence, clinical observations?
I think this is urgently needed. I mean, I guess for PTH, we do have already some evidence, particularly from primary hyperparathyroidism, that indeed can lead to symptoms similar to those seen in patients with chronic kidney disease and mild cognitive impairment. So I think this is a very strong indicator that we should go in that direction. And for phosphate, I think the evidence at the moment is not there, but this could simply mean we haven't looked yet. And I guess it's really worth to be looked at. Of course, both PTH, the rise in PTH and the rise in phosphate are major targets for nephrologists to treat. And so if we could use these parameters also to prevent the cognitive decline in the patient, I think this would be very important.
Okay, we are running out of time for this first speaker. I see another questions from our audience, and we will take the question at the end of the three speakers. So I ask now my co-chair Marion Pepin to introduce the next speaker. Thank you.
Yes.
Carsten.
Thank you very much, Carsten, for your nice presentation, and thank you, Gianni. I'm pleased to introduce the next speaker, Julie Refardt. She's an endocrinologist with a patient for saltwater disturbance and a special interest on hyponatremia. So she will give us a talk about the role of sodium in the brain function. Thank you, Julie.
Yes, thank you very much, Marion, for the kind introduction. And thank you for the invite for giving this talk about one of my favorite subjects. I hope you can see the slides okay. Okay, so because I could talk for hours about this, but I have to limit myself to 15 minutes, I would like to give you a quick overview about the causes of sodium disturbances. Second, I would like to talk about the effects of sodium changes on the cells and brain cells. And then I would like to talk about the effects of chronic hyponatremia on brain function and if treatment has a reversible effect on this. And then I would like to summarize my talk. So, as you know, the causes of hypernatremia are quite diverse. So the most common one, which you're probably all familiar with, is volume overload in decompensated heart failure, kidney failure, or liver failure. The most easy to treat, in my opinion, is the volume deficit. So if a patient loses volume in gastrointestinal loss or other losses. And my personal favorite, the syndrome of inappropriate antidiuresis, where hypernatremia is caused because of the free water excess. There are also other causes which are in between. So we have the primary polydipsia, we have free water excess with volume overload. So hyponatremia is quite a complex disease. It takes a detective mind and gives most physicians a headache to diagnose it, but it's also very exciting. Then if you look at hypernatremia, that basically just includes not having enough water. So on the one hand side, maybe you have a patient who does not have adequate access to water because he's immobilized, or because he's delirious, or because his first center does not work correctly. Or again, you have loss of water, so if hypertonic fluid loss, or you have a defect at the kidneys of the concentration defect due to diuretics or nephrogenic or central diabetes insipidus. Or rarely you have excessive salt intake, usually iatrogenic to physicians administering hypertonic fluid infusions or parenteral nutrition. So what are the effects of sodium changes on cells? So as you know, the cells basically swim in salt water, and they would like to keep this homeostasis. They would like to keep their cell sizes at all time. So what happens if you look here at the left-hand side with hyponatremia, that usually induces hypotonicity, and that causes the cell to swell. And if that happens too quickly, the cell has no chance to adapt to these changes, that leads to to breaks in the cell and finally to apoptosis. However, in a chronic state of hyponatremia and therefore hypotonicity, the cell can adapt, and it can do so by losing osmolytes, for example, glutamate or taurine. By losing those osmolytes, it can keep its size and therefore prevent its apoptosis. On the other side, if you look at hypernatremia here on the right-hand side, that leads to hypotonicity and the cell shrinks. So if the cell shrinks too quickly, again, it cannot adapt. It leads to damages on the cytoskeleton. It leads to breaks in the DNA, and again, the cell dies eventually. Of course, the cell tries to prevent this. So if we have a chronic hypernatremia, it can again try to counteract this by taking up osmolytes, again, glutamate or taurine, and therefore keeping its normal cell size. So this is, of course, oversimplified, but that's the basic mechanism you have to keep in mind when talking about hypo and hypernatremia. So why is this important, especially in the brain? Because the brain has the unique limitation that the cells can only swell and decrease to a certain limit because it's in the brain. So what happens if there's a rapid onset of acute hyponatremia? If there's cell swelling, it comes to a cerebral edema. Your patient will have severe headache. They can be delirious. There can be vomiting. Worst case, they can have seizures, be in a coma, and in the really worst case, it can come to herniation. On the other hand, with a rapid onset of acute hypernatremia, again, we have the cell shrinkage. So this can lead to blood hemorrhage in the brain. And also because the astrocytes, you can see here, they try to protect the neurons from quick changes in the tonicity, so they can be damaged and it can come to the osmotic demyelination syndrome. So the problem is in chronic disorders where, say in chronic hyponatremia, where the brain tries to protect itself against the swelling, if you then correct the hyponatremia too rapidly, you can also induce osmotic demyelination because the brain cannot react as fast to correct these adaptations, because it can lose the glutamate and the taurine, but it takes much longer to take that back up into the cell. So if you treat hypo and hypernatremia, it's very important that you keep in mind how fast those changes came. So if you have acute hyponatremia, so if you have a patient who is severely symptomatic, has a decreased sodium level, you should not lose time. So you should treat them with a bolus of hypertonic sodium. Usually, one bolus increases the sodium level around 2 millimoles per liter. You can repeat those boluses 2 or 3 times because increase in sodium level of 4 to 6 millimoles per liter is often enough to correct the acute symptoms. Similar with the treatment of acute hypernatremia, just give them volume, try to decrease the hypernatremia quickly. However, as we discussed before, in chronic hyponatremia, you have to be more careful. So whenever you're not sure if it's acute, then it's usually chronic. And also if the patient is not severely symptomatic. And here it's recommended that you avoid to increase the sodium levels above 8 millimoles per liter, because as we said, the cells need time to readapt at the changes in osmolality and sodium levels. And also you should consider to relower it in case they increase too fast. Treatment of chronic hypernatremia, it's a bit less studied in the literature. Usually in children, you're advised to be more careful. In adults, actually, we just say we take around the same correction limit as we have in hyponatremia, and you do not decreases above 10 millimoles per liter. But in my experience, actually, those patients are usually under treatment undertreated because physicians tend to underestimate how much loss, how large the water deficit is in those patients. So it has been debated for a long time if mild and moderate hypernatremia is really symptomatic. And I feel confident to tell you it is, because we have a lot of observational studies and also some interventional studies who showed us that there are effects on the general well-being, quality of life, neurocognitive and neuromuscular function of the body. And here on the left-hand side, you see the results of an evaluation we did in hyponatremic patients. We asked them about specific symptoms common with hyponatremia, like headache, confusion, tiredness, forgetfulness, and they were quite frequent in those patients. And also after treatment for only 2 days, most of those symptoms decreased, even though we were not able to normalize the sodium levels on many in those patients. Similar results were seen in several tolvaptan trials. So vaptans, you probably know, vasopressin antagonists, so they lead to aquaresis, therefore correcting hyponatremia quite efficiently. And there was a study in 24 hyponatremic cirrhotic patients, which they saw that tolvaptan led to an improvement of the general mental health. And the same was seen in another patient with U and hypovolemic patient, where also an improvement of mental health was observed. So how about the effects of hyponatremia and treatment effect in neurocognition? So here it is a bit less clear. So we do know hyponatremia has an effect on the neurocognition. Here on the left-hand side, again, a study from our team in 19 hyponatremic patients. We had to do several tests on the computer, testing the attention and reaction time. And we saw that the hyponatremic patients had a lower alertness, had a shorter attention span, and a slower reaction time and higher error rate. Similar findings were seen by the Belgian group, Renebourg et al., which they also saw in 16 hyponatremic patients the same slow reaction time and higher mistake rate. Interestingly, when we looked if treatment improved this function, we only saw that in a subgroup in the go, no-go test. And the similar findings were seen in the INSIGHT trial by Giovambattista, which also looked at again at tolvaptan. They also saw no difference in neurocognition after the treatment, except in the subgroup of the psychomotor speed domain. So here, it's not quite clear why we do not see a stronger effect of hyponatremia treatment on the neurocognition. One factor it could be is that we're not using the most sensitive tests. And here we did an ADIUS study we just recently completed in 16 outpatients with mild to moderate hyponatremia. And we used the Montreal Cognitive Assessment Test, also called the MOCA test, which is known to really be a very sensitive detection of neurocognitive impairments. And we saw that after 4 weeks of treatment, we saw an increase in the MOCA score in the normal range. And this was also during a subgroup analysis of executive function. And here on the left-hand side, you see an example of a patient before treatment, which she was hardly able to draw this cube and also a watch. And later on, this actually looked a bit better. So for the neurocognition, we still do not know what is happening here. We seem to have some effect, but not always. And we also need further studies to evaluate why this could happen. And then what about the neuromuscular function? So this, again, the study by the Belgian team, Renebourg, which saw impressive results in so-called asymptomatic hyponatremic patients, which had a massive increased rate of falls of 21.3% compared to a group of matched control patients. They then took a subgroup of patients and did some gait analysis test. You see different severities of hyponatremia 130, 126, and 124. The patients had to do those tandem steps and the error rates, and then you see again the patients after treatment where we see much better results. However, also here, other studies which also looked at the gait did not see an improvement here. And in our study I showed you before, we also did different gait tests and we did not see a change there. So again, maybe it needs longer time, maybe we do not see everything there, so we need further studies. One factor which could be is that because the cells have to lose glutamate to keep the cell regulation going, maybe this could also have an effect on these neuromuscular, neurocognitive disorders, and maybe this could be a cause why we cannot correct it as quickly as the hyponatremia itself. So this brings me to the end of my talk. I hope I was able to show you that sodium homeostasis is of eminent importance for the brain function, that changes cause osmotic stress leading to clinical symptoms, that the brain has the ability to adapt to abnormal sodium levels. However, but the readaptation needs time and also bears risk of additional symptoms, that chronic hyponatremia is definitely not asymptomatic, and that treatment of chronic hypernatremia is only partially efficient in reversing those symptoms. And with that, I would like to thank you very much for your attention. Here you see a picture of our research group. If you're interested in hypernatremia, please feel free to follow us on Twitter. And now I'd be happy to answer any questions. Thank you.
Thank you very much, Julie, for this super didactic talk. I think it was very clear. And as we can see in the question, we can see that it's a really important question for physicians maybe, and because it's involving chronic kidney disease and aging people, which is really frequently questioned in this field. We have several questions, but maybe I would choose one because we are to stay on the time. Do you have something to tell us about the limit for the physical adaptations of the changes? The person is asking about the different electrolytes, but maybe you can answer for sodium. And I think the question is about what is there a timeline where the body can adapt, and is there any limit that you can tell us for adaptation, for physiological adaptations?
Well, that's actually a very good question, and I don't know. So because all those studies I showed you, they had different observational times, we also thought that maybe it's a question of time. So we extended the observation up to 4 weeks, but still then we did not have a complete remission. So actually it seems to take longer, but how long exactly, I cannot tell you. I also think it could be quite individual from patient to patient, how long this time needs, and also depending on the underlying cause of the hyponatremia itself.
And in the same question of the time, do you think for clinician that there is a time that you can spend under the limit for natremia, for instance, that will have consequences on the long term? I think I saw some papers about the past year average sodium on hemodialysis patient that was linked to depressive symptoms or cognitive symptoms. So do we think that it's acute or a chronic dysnatremia, which is the problem, or both maybe?
I do think the chronic is the problem, because the acute, their patients are usually quite symptomatic, and this also gets treated quite aggressively. And I think the problem is the chronic hyponatremia, which most physicians still think it's just an innocent bystander and does not deserve treatment. So I think there it's really the chronic that is the harmful one.
Thank you very much. Is my co-chair okay? So we go to the next presentation.
Yeah, you can go on.
So my pleasure to introduce Pedro Henrique Imenez Silva, who is a biologist and renal physiologist working on the role of acid-base balance in renal pathophysiology. And I think he will give us a presentation about acid-base and brain function.
Thank you for the nice introduction. Thank you for the invitation. I think it's a wonderful opportunity to discuss these complex interactions between kidneys and the brain. And now we've just heard about phosphate, about sodium, and now I'm going to bring this acid-base physiology perspective to this discussion. We've heard from the previous talk, so I don't have to talk much about it, how there is a high prevalence of declining in cognitive function in patients with chronic kidney disease. However, when we have to take in consideration the role of acid base as a potential intermediator between kidneys and brain here, we have to take some theoretical aspects in consideration because we don't have actual experimental demonstration that acidosis could be this link between the chronic kidney disease and the cognitive impairment. So what I'm going to cover in this presentation, there's 3 questions, and some of them you're going to see they are reasonably open. First, is there an association between serum bicarbonate and cognitive function? Serum bicarbonate normally used to define metabolic acidosis. Can acidosis secondary to chronic kidney disease cause acidosis in the brain? And what are the effects of acid-base disorders finally on the brain? So jumping directly to the studies that tested for the association between serum bicarbonate and cognitive function, you can see that the list is not very long. Sorry for the wall of text here, but we have only 4 rows for these works. And it's also hard to identify because not every work clearly basically show the bicarbonate data that they have there. But here on top, we show these two studies, one with hypertensive patients with and without chronic kidney disease, almost 3,000 patients in the United States, and another work with 190 patients with chronic kidney disease in Nigeria. Both of them showed association between low bicarbonate and reduced global cognitive function. In pediatric patients, with almost 900 patients here, it was not found an association between bicarbonate and executive functions. And in 65 hemodialysis patients, there was an association with lower sleep quality, but not with other cognitive functions and depression. There are some also other studies related to gait speed and lower extremity strength. And there is also a relation between bicarbonate and these parameters. So the bottom line here is that we don't have many studies to base this. It seems that there is an association between bicarbonate and cognitive function, but these studies evaluate different cognitive domains. They have different cutoff values for bicarbonate to define metabolic acidosis, patients with very wide spectrum of GFR. So it's definitely necessary to have more studies to identify if there's indeed an association between these parameters. So, but let's look at more in the mechanistic perspective, how this could basically be happening. One of the first ways that you could look at it to have potential insights is to look whether the prevalence of mild cognitive impairment would be similar to the prevalence of metabolic acidosis. If they were very similar, potentially they could be happening in parallel or they could be interacting. When we see here the prevalence, the typical prevalence of metabolic acidosis and in patients with chronic kidney disease, you see that they are quite different. So it's basically not very prevalent in the early stages when the kidney function is high, but much higher than the prevalence in late stages. This, if we use the definition of serum blood bicarbonate or total CO2 below 22 millimolar. The question is that this definition of metabolic acidosis may not be enough to understand what's happening to these patients. This because more recently it has been proposed a new theoretical construct which identifies metabolic acidosis in a bit different way. In early stages of chronic kidney disease, patients would have this plasma bicarbonate within the normal range. That's exactly what we see here in the graph on the left, right? But these patients could already undergo an accumulation of acids in the intracellular space and in the interstitial space. And then with lower kidney function, there is a higher accumulation of acids. And only after a certain level of accumulation, and in general with more reduced kidney function, we would see the bicarbonate levels really reducing below the normal range. Therefore, we call this situation as a preclinical, subclinical form of metabolic acidosis as eubicarbonatemic acidosis. So this eubicarbonatemic acidosis is in contrast then to this metabolic acidosis that in general is used to measure metabolic acidosis clinically. The problem is we don't have yet any idea what's the prevalence of this eubicarbonatemic acidosis in patients. We still need to develop also more tools that are easy to evaluate this condition in patients. So taking now this in consideration, we can see that they could be interacting in early phases when we are not seeing that this metabolic acidosis is indeed there. To understand then this relationship, we have to understand how fluids are distributed in the brain and the central nervous system. Typically, the samples to measure the pH in the brain are taken from lumbar punctures of the cerebrospinal fluid. The CSF cerebral spinal fluid is produced by the epithelial cells of the choroid plexus, which are then in constant exchange with the blood and the CSF in here. This forms then the blood CSF barrier, which is less famous than the sibling here, the blood-brain barrier that we have in here, which has an endothelial cells in between the blood and the brain cells and the interstitial fluid. It's important then to recognize that in these barriers, several solutes simply cannot cross freely from the blood to and reaching finally the neurons and astrocytes because of these barriers. The paracellular, especially the paracellular transport here cannot allow the easy transport of bicarbonate or of protons. We have multiple bicarbonate transporters that can do this, but this is a much slower movement, while CO2 can diffuse through these barriers. This is important to understand the relationship in acid-base, because which can be quite nicely illustrated by the condition that was shown by Postnatal and Plum in the New England Journal of Medicine in '67, where they tried to identify patients that had severe cases of metabolic acidosis. And then they looked at patients that had received bicarbonate infusions, either only bicarbonate or bicarbonate with insulin and some other substances. Here we have blood pH, which is quite acidic, below 6.9, and then bicarbonate is given to these patients, and as expected, bicarbonate raises. But look what happens to the CSF pH in these patients on average. Actually, it was reduced. And this is expected because CO2 now, because it's increased the total CO2, can rapidly cross the barriers and finally reach the CSF in here. So therefore, you can have an acute acidification, which is paradoxical if you think why this patient has an increase in the arterial pH. So we have to take into considerations these differences when we compare simply pH in the arterial blood outside of the brain, and then when it is in the brain. So how does it actually look in a more chronic condition? Because when we are talking about chronic kidney disease, you are talking about also chronic metabolic acidosis. In this condition, this was evaluated in the 60s, and here is an example with healthy subjects. So these were four, typically as at the time, so young male subjects. They received ammonium chloride to induce acidosis or sodium bicarbonate to induce alkalosis. Several determinations were done with these patients, and successfully they reached acidosis with ammonium chloride and mild alkalosis with sodium bicarbonate. But look what happened to the CSF pH of these individuals. They had a reduction and an increase, but if the difference here was of 0.1, 0.15, here we're talking about 0.01, 0.005 of a difference. Two different methods were used to calculate the CSF pH, so they have different absolute values here, but the pattern was the same. So how does it look for chronic kidney disease patients? 1 year earlier, it was published in the same journal, and authors here, they collected several different conditions that would cause acidosis or alkalosis. So I cropped a little bit this table here just to show the line with chronic kidney disease, and another one when patients were receiving chronically sodium bicarbonate for 1 year. So arterial pH was reduced in relation to the healthy patients here or with normal conditions. And it was increased by sodium bicarbonate ingestion after 1 year, so as expected. And CSF, again, there is a same direction of change with reduction in CSF pH in acidosis, an increase in alkalosis, but these differences are again much smaller, so it's highly attenuated. So the conclusion that we have in here is that the brain is highly protected from these changes that are happening in the arterial pH. However, there are still changes and they go in the same direction. The question is, can a very low-grade acidosis, in the case of eubicarbonatemic acidosis, cause small changes with an impact in brain function? So for this, we have to look at the data of how the CSF pH changes brain functions. And here in AL69, in this paper, it was measured ventilatory response in humans and cerebral blood flow also in humans. And you can observe that these are very steep associations. So the regression line is not here, but it's very easy to see how steep it is. Like a difference of 0.05 is already enough to increase the ventilatory response in 8 to 10 times. The 4 small changes in pH have a high power to change brain function. Still not clear whether eubicarbonatemic acidosis could cause these changes. But of course, with in later stages of chronic kidney disease, this acidosis is accumulating and then is expected to impact CSF pH and therefore brain pH. When we see that mechanisms overall and how acid-base and acid-base disorders impact the brain, I brought here a few examples. So I told you about cerebral blood flow, about ventilation, but also there are important changes in cell energy metabolism. This is something that has been more recently investigated. And also for quite a while, we know that pH changes neuronal activity. So there is again potential to change brain function. We can look at also not only locally, but systemically, because pH changes the activity of immune cells, and therefore this could also have indirect then impact on the brain. And acidosis also interacts with a multitude of substances, of circulating substances that could also potentially be involved in several conditions associated to cognition. So we have renin-angiotensin aldosterone system, clotho-endothelin, catecholamines, and so on and so forth. I'm going to just cover a single example here because I find this is a quite actually beautiful example of how the physiology of the brain is interacting with acid base and also with mood-bothered activities. So there is the astrocytic neuronal lactate shuttle theory that says that here is a neuron on the left that when it has a higher energy demand and higher energy expenditure, there is a communication that very quickly with astrocytes increase the glycolytic activity and then exports lactate directly to the neurons. And this lactate can be used as a fuel for the activity of the neurons. What has been more recently published is that this higher activity of the neurons that are again here on the left also acidifies the milieu. So this is a microenvironment around the neurons and the astrocytes. At the same time, then the astrocytes secrete bicarbonate, neutralizing this export of protons, therefore showing that concomitantly we have this higher energy expenditure, but also bicarbonate secretion. I can only imagine that a local acidification could also change these neuronal activities. So to summarize, acidosis now it's known to impact kidney function in itself and then change in CKD progression, which then could indirectly impact brain function. Now, our question is whether acidosis can directly affect the brains beyond also what we know and is more recent in interactions with systemic inflammation and also with the muscle activity. We need then to answer a few other questions, and we need now that the alkali therapy clinical trials evaluate cognition. We need better tools to measure small changes in local pH, And we needed an assessment of brain acid-base status in chronic kidney disease animal models, and of course, later on, we live in humans as well. And then to finish, I just would like to acknowledge the Connect Action and all our colleagues that helped with this work, and also the collection of the data, and also the funding agencies. Thank you very much, and thank you for your attention.
Thank you very much, Pedro. I don't think we have specific question on acid-base regulation, but I would like to ask, what do you think, which brain function would you focus on? Which brain function do you think would be the more impacted by acidosis, or is it cognitive function, or is it could impact seizures? I don't know if you have had a look on electrophysiology impact on, I think there's things.
Yeah, so I think this is a multi-level question, because we can talk about what is happening to neurons and to cells, and also in cognitive domains. When we talk more in the macro level in terms of cognitive domains, we unfortunately don't know much right now. There are these works just basically showing like global cognition that's reduced and potentially executive performance being reduced. And we have a little bit more works is about like reduction in gait speed, reduction and in strength. The problems that this can also be neuronal, but there is a direct impact of S in muscles. So, and also in catabolic activity. So it's hard to differentiate. When we go directly to the cells, I think it is now quite clear that pH changes the activity of neurons. And while there are a few works that shows that acidosis, for example, causes excitotoxicity in the neurons, I think the large majority of the works have shown a reduced activity by acidosis and an increased neuronal activity by alkalosis. Therefore, we have both sides of the spectrum of acid base that can change directly neuronal activity. And astrocytes are also behind like good part of those performances, because I didn't show this or didn't describe this part, but in the blood-brain barrier, we have as if it was a wall of astrocytes that are secreting constantly bicarbonate, and since that they break good part of the changes that could be coming from the blood in there. So I can only imagine that having such a strong break system for the changes from the arterial pH to brain, that this can be really sensitive to pH in multiple levels.
Marion, may I ask a question to this very, very interesting talk by Pedro? Really fantastic, really. Actually, we understand now how the brain can protect itself from change in the pH. Is absolutely marvelous that there is a microenvironment done by the astrocyte in order to protect the neurons and the connection with the other neurons. So it's a fantastic relation, a very, very well to study. But you know, when we start with the connect activity, it was clear to us that actually the decline in renal function was somehow correlated with a decline in an impairment of cognitive capacity. And that now you have explained that actually that there's this correlation can be somehow be straightforward, because if we consider not bicarbonate but other determinants for the pH status of the brain. But actually, what I want to tell you that there is not a model, a status of patients when we can look abruptly to change in pH. And these are the patients that are under dialysis. You know, under dialysis, there is, no matter what, a very high decline of cognitive function. And during the dialysis, the patient goes from profound metabolic acidosis to metabolic alkalosis because they receive a large amount of bicarbonate. So actually, there is with the possibility to look at the brain function under condition of metabolic acidosis and metabolic cirrhosis. So it will be, let's say, a way where we can study the action of a change in pH. The point is, this will be 2 short time observations. And so probably we don't, since the brain can keep a kind of autoregulation pH, will we be able to look at that or not? So this is one question that we can pose to the connected people, I think.
Yeah, absolutely, I agree. I think this is something that could be looked at it, and there is only this single work with, I think, 65 patients under hemodialysis, and it was not found an association between cognitive function and bicarbonate in these patients. However, we need more studies. Just basically, we need more studies. I think the potential of pH and acid base overall being directly connected to cognitive impairment is still very theoretical. Like we have now the tools, we have the information, but we need studies now to identify these conditions. I absolutely agree.
May I pose one question to all the 3 speakers, Carsten, Julie, and Pedro? You know, when we speak about cognitive impairment, we speak of many subdomains that are somehow the target. Do you think, Carsten, Julie, and Pedro, that there are a specific subdomain of cognitive impairment that are affected by phosphate, by sodium, and by pH?
I don't know who's to start. So then I start maybe because I talked first. So for phosphate, the short answer is we don't know. There are no good studies that really have looked at the association between phosphate levels and cognitive functions, and so there's of course no subcategorization here for different domains. So what has been studied much more is really the vascular risk for the brain in patients with hyperphosphatemia, which I think is logic given the high cardiovascular risk in patients with chronic kidney disease. And of course, there is a vascular form of dementia. So it is tempting to think about this, and but it really needs studies. And I guess what will be difficult to some extent is that the change in phosphate in patients with chronic kidney disease comes not alone. It comes with a myriad of other uremic toxins, with changes in acid base, in changes in endocrine factors. And so it is probably not very simple to dissect single factors out of this toxic cocktail.
Julie, anything to add? Because I have for you one in addition to this question, because it's a very astonished that actually when we correct all the studies where have been corrected hyponatremia, they have not been reported an effect on the total cognitive capacity. Probably one reason would be that what you pointed out, that it would be too short in order to see change in cognitive capacity. If we related to the sodium level. Another possibility that actually they have not looked at a specific one of the subdomain of the cognitive capacity. So
exactly, yeah. Thank you for this question. That's really interesting. So what I can say that you see in hypernatremia, a general decline on neurocognitive function. However, it's really unclear which part is really affected by the hypernatremia, because those patients are usually comorbid, they're sick, they're often elderly people. And we only have limited results from actually interventional studies. So most results we have from observational studies. And the limited evidence we have from interventional studies that it seems the executive function seems to improve. So that's what I can tell you here. So this is certainly something we will look further into, and it's a fascinating subject. What is the cause and what is the consequence?
In terms of pH, it's not known either. So we don't have even the advantage of having the interventional studies that could bring us. Bicarbonate could be just a consequence of better or worse kidney function, and of course also some dietary factors too. So it's not known yet. There are the few works just looking at maybe a bit of executive performance, and there is association with bicarbonate and sleep quality, but not much more than this actually is known right now.
Marion, I think there is one question from the audience about German immunos. Do you want to?
Yes, I think it's more a question for Julie.
Yeah.
The audience is asking if you can say what is the level for the serum hyponatremic chronic that will have symptoms. Is there a cutoff where you can say some symptoms are coming?
Yes, so a long time people thought only severe hyponatremia makes symptoms, but there have been a lot of studies that show even mild hyponatremia. So even between 130, 134 make symptoms. So there's really no safe cutoff. And I think that's quite interesting. And it's also shown that even mild hyponatremia is associated with increased mortality. So I think probably mild is the wrong term for this disorder.
Do you have any further question for the 3 speakers?
I wanted to, as a physician, I wanted to ask the 3 speakers in each of their field if they would like to say to the physician what we should pay attention on. For instance, for the phosphate balance, should the physician be more on the phosphate level or on the PTH? Because we discussed that we have consequences with PTH and phosphate. For natremia, you said that it was about chronic natremia, but maybe you want to add something for the physicians. And for Pedro, because you said that the acidosis is not a good reflect of the acid accumulation during CKD. So maybe you want to say what the physician can pay attention on.
Okay, so again, for phosphate maybe first. So I guess in patients with hypophosphatemia, I guess that first of all, the underlying disease should be identified and treated. And of course, hypophosphatemia often comes as an incidental finding, and only if it's really very severe, it is symptomatic. In patients with hyperphosphatemia, this is usually associated with chronic kidney disease, or is the consequence of chronic kidney disease. And then I guess it's anyway a multi-level approach to these patients that includes usually PTH. The story is certainly a bit different if there's an isolated increase in PTH, like in syndromes of primary hyperparathyroidism. And then, of course, the cause of the hyperparathyroidism should be corrected because it has not only consequences for the brain, but of course also then for bones and calcium levels. And I think it's the more important, at least from the perspective of the brain, because it seems that cognitive dysfunction and mood disorders can be corrected and can be reversible with correcting PTH levels. So there is really a therapeutic benefit for the patient.
My message for the physicians would be don't ignore the hypernatremia. I know it can be brain wrecking to see where it comes from, but please take the time, go to the trouble, look at the urine, look at osmolality, see where it's coming from and try to correct it, because I believe you will, this will have a benefit for your patient.
Yeah, in terms of acid base, so first take with a grain of salt. I'm not a physician, so this is the biologist telling. There is a study that tried to identify the percentage of patients who would be eligible for correction of acidosis. And so basically, what's the proportion of these patients that are receiving correction for acidosis, for example, with sodium bicarbonate, potassium citrate, and so on. And the percentage was very small. It's very, very, very small. So it seems to be clearly a neglected issue worldwide among physicians, especially nephrologists tend to look at several other aspects related in this complex thing, which is the chronic kidney disease. And often acid base is just left behind. The problem now is, if the still theoretical construct of eubicarbonatemic acidosis is correct, and I believe it is, how do we identify the patients that are under a higher risk or are accumulating more acids? And we don't have tools yet for this. We have some proposals, like the one that has been proposed recently is to measure urine citrate, but it's not a thing that it's very easily to be measured in any hospital at any time. So basically, I think we will still need to develop tools, and tools that either by or imaging in the blood and in the urine that could quickly identify those patients. And then we also need clinical trials that then, when they are identify those patients, that you can give the correct dose for the correction of acidosis. So it's still quite a complicated issue that has to be solved.
Well, you have been 3 marvelous speakers, I say, and now I would like to leave the words to my co-chairman to conclude this. I think it is very, very interesting, although more experimental than clinic. Symposium, because of the things we have to know, they are much larger than the things that we know. If you want to conclude.
Yes, I think
Marion?
we can. Yes, thank you. I think we first can thank our 3 brilliant speakers, because it was really interesting, and the link between kidney function, electrolytes, and brain was demonstrated. And so now we can see that we have a lot of work to do, a lot of clinical studies, a lot of biological studies. So it's a good point for us to know that we have many things to do. And I think we will have also interesting sessions during the Congress about the link between kidney, electrolytes, and brain and cognitive function. Thank you very much.